Hi, my name is Kim Adler. I'm a technical product owner at Pfizer, and I specifically sit in the digital organization. So the digital organization creates software and digital tools to help facilitate operations at Pfizer, and we specifically support global supply. So that includes supply chain, engineering, quality, etc. And within our group, we've developed a knowledge graph database for operations data, which has over 1 terabyte of data. So we pulled in data from a data lake that contains information on quality, investigations, manufacturing of batches, as well as unstructured documents as well. And we've all integrated that into a graph database. And what I'm going to go through here is a use case that has leveraged this knowledge graph. And it's for a specific tool that's meant to identify chemicals of concern used within Pfizer products. And there's my first slide. So here's some context. So earlier this year, the FDA revoked authorization for the use of red dye number 3. Is anyone here familiar with chemical bans? Right, so when this happens, we have to act very swiftly. And this requires not only identifying where we use these chemicals of concern, but eventually we have to figure out a way to replace them in our manufacturing. And why is this such an important issue for us to address? Because this is something that is happening all the time. So red dye was just the most recent case, but in the past several years, we've had to deal with identifying a bunch of different chemicals of concern, like all these different dyes, DEG egg, PFAS chemicals, for example. So what's our goal? Our goal here is to identify all commercially manufactured products containing any chemical of concern efficiently. So that means not just having it in a very performant, quick way, but something that's generalizable, right? Because chemicals can enter at various stages in the manufacturing, and the products that we manufacture are very diverse. So we manufacture small molecule products, large molecule products, and they have very different recipes. So we need a solution that can efficiently generalize to any of these types. Next, we have to do this completely. So this is very important. We have to make sure that we don't overlook any instances of a chemical of concern being used in a product. Not only does that make us not compliant, but that's ultimately harmful for our patients. So it's extremely important that we're able to identify every single instance in which a chemical of concern is being used and then address that swiftly. And then lastly, we have to be very interpretable in our understanding of where chemicals are being used. So it's not just sufficient to say, okay, these are the products that we sell that contain this chemical of concern, but where does this chemical of concern come into effect? Because we need to be able to ultimately address how we're going to replace that chemical. And so this is an example of how we address this using graph database. So this is our starting point here. So we have a big data lake of tabular data. And one of the tables that we have is material movements data, which is essentially an aggregation of batch level movement data. So batches are instances of materials, and we use them to then as an ingredient into a downstream batch. So this happens over and over and over. And this data is initially stored in a tabular format. So I've anonymized everything. So of course, we don't have a chemical named name 1, but you can imagine it being benzene, for example. And what we have here is an identifier for the material, its name, what type of material it is, so if it's a raw material or a packaging material, if it's a semi-finished API or a finished drug product. And then we see in all instances of this material, what has it then been used to manufacture downstream. So in the first record, we can see that name 1 was used to then produce name 10, which is a finished product, and that is manufactured at site C, and so on and so forth. So we could say have another raw material, and that instead of going directly into a finished product, it goes into a semi-finished API, and then the semi-finished API goes into a finished API, and so forth. So if our goal is to ultimately understand which finished drug products, like what we ultimately sell to the consumer, contain a chemical of concern, looking at this data, is there anything that jumps out to you that may be slightly problematic or challenging in using data in a tabular format? If you don't know, I'm about to explain it, but does anybody care to wager a guess?
You could have many different levels, so it could drill down many levels before we get the whole answer.
Yeah, absolutely. And that's exactly what I'm going to go into. So in this first case, right, we see that we have the raw material is going directly into the finished product. So each record in the table is just one material movement. So you have the input and then the output, right? So in this case, because the raw material feeds directly into the finished product, we're good, right? This one record in the table gives us all the information we need to know. But as she mentioned, oftentimes, these chemicals don't directly feed into the finished product. They may feed in very early into the genealogy or the recipe for how to make this product. So here we see name 2, say that's a chemical of concern. Well, if we just looked at the one record in which name 2 exists in our table, it feeds into a semi-finished API. And we don't know ultimately what that finished product is. So it's not sufficient to just look at each record in the table and know where all of our chemicals of concern are being used. And now think about it. How would we ultimately derive the relationship that name 2 is indeed used to manufacture name 10, the finished drug product? Right? It becomes very difficult and very manual because we honestly don't know how many hops away the raw material is from being used in the finished drug product. So ultimately, what we'd have to do is for every single time a new chemical of concern comes in, we'd have to continuously join on the input material and the output material until we hit on a finished drug product. And I specifically and intentionally put in ... there because we do not know from the outset how many hops away a raw material is from a finished drug product. Imagine just the nuances between a small molecule product versus a large molecule product. Their genealogies are completely different. So working with tabular data in this case becomes very manual and difficult to use. But we're able to resolve this issue by structuring this tabular data in a graph format. So this is the turning point for us. So let me step you through to this graph model. So we see here we have our materials, and we reflect them as nodes in a graph. And then we have these ingredient to relationships. And you can imagine these ingredient to relationships capturing each row in that table, right? So remember we had the first row in the table here. That's reflected by this relationship here. Name 1 is ingredient into name 10. And then this is very effective for when chemicals occur very early on in the genealogy. Rather than having to do those joins every single time a new chemical comes in, we solidify all those relationships in a graph format. So we see here we have name 2 is an ingredient into name 3 is an ingredient. So it's literally saving each of those records together as a node relationship node pair. And then on top of that, you can layer in additional relevant data. So for example, we saw in that table, we had information on where these materials were produced. You can layer on top of that site nodes, for example, and say, these materials are produced at this site. So why is this so helpful for us? Because we've named these relationships in the same way as ingredient to, all we need is one simple Cypher query to surface all of the downstream materials in one shot. And we don't even need to know from the outset where that chemical lives in the genealogy. You see here we have this asterisk here, which basically says variable number of iterations for a relationship. So it captures both the use cases we were talking about before, where in this case with name 1, it's only one ingredient to relationship. But here we have, 4, 5, 6. It doesn't matter. Just by running the same query and plugging in the name, so instead of name X, you plug in name 2, this works for every single use case. So now that we've covered the whole efficiency, generalizability issue, now we have completeness. So I'm sure, as all you know, chemicals have very different naming conventions and different aliases. So what I did here was I took the initial slide, I went into PubChem, and I listed all, like a subset of all the different ways these chemicals are referenced. And as you can see, they're completely different from each other. I mentioned FD&C red number 3, that's red dye, but it's also known as erythrosine. Keyword searching here, if I just search FD&C red number 3, we're going to overlook all instances of these synonyms. And even with, say, regex, for example, if there's like an extra space, for example, we maybe will still be able to capture FD&C red number 3. But something that's referenced as erythrosine, which is a completely different spelling, we're going to overlook those entirely. And in terms of this space of compliance, when it comes to the health of our patients, we can't overlook any of these instances. So I'm going to pause here and ask you guys, what approach would you take to try to address capturing all these different variations of a particular chemical? And if you don't know, that's fine, because I'll tell you the approach that we used.
I would use graph.
Graph? Yeah. Graph is definitely part of the solution, absolutely. Yes? Different properties, yeah. If we knew from the outset we could store all of these different names as a property in the graph, like a list of aliases, and that's something we have done. Any other ideas?
Make those aliases nodes to also known as.
That's another great one. Well, I'm going to go and hop on the Kool-Aid train and say we can instead use AI and large language models. And what all of you guys have said are completely valid solutions if you know from the outset all of the different naming conventions, right? But sometimes you may not. And imagine if an entirely new chemical comes in, you'd have to, from the outset, manually list all those different variations. What you can do is you can leverage a large language model that has been trained on biomedical data. So we talk a lot about GPT-4o, for example. But in this particular case, because we're dealing with a very specific domain like chemicals, you can actually use smaller language models that are trained on a much more relevant set of data and run this locally on your computer. So one of them is BioBERT, right? This is a large language model that's trained on biomedical data, for example, PubMed data. And you can use that to encode each of these material names and then leverage semantic similarity to return not just the exact match, but all of the similar matches. And so what I've done here is I've created a visualization for how vector embeddings work. Obviously, with a large language model, the number of features is in the hundreds and the thousands, so that's not something you can really understand by looking at it. But here I just, for example, am showing in two-dimensional space how we can see how these different synonyms are similar to each other using vectors. So for example, here we have FD&C yellow number 5. It's not the exact same vector as tartrazine because it's not the exact same word. But if we used a large language model that's trained on PubMed data that's seen many instances of these different chemicals being referenced, it understands that it essentially means the same thing. So we can see tartrazine points in a very similar direction as FD&C yellow number 5, right? So you can use the relative distances or angles between vectors to extract not just the exact keyword searches, but all of the different synonyms. And you can do this easily using Cypher. Right, so I've added an additional property onto these material nodes. So what we have here is we take our name property and we say pass it into a name embedding LLM. So I try to illustrate that here with this kind of red text as a function. You can imagine it being BioBERT or you can imagine it being GPT-4o. And you save that in the graph and then you put an index, a vector index on it. And that makes the search a lot more efficient. And then say you get a chemical that comes in like name 1. Remember in the previous query, we searched on the match where name is equal to name1. Instead of name is equal to name1, we're passing in the name embedding LLM. We're passing name1 as a parameter, and we're getting out a sequence of numbers. And what you can do is you can use different similarity metrics to then surface the most relevant materials. So here I use cosine similarity, so that literally looks at how close the angles are between two different vectors. So if vectors point in the exact same direction, they have the exact same meaning. And what you can do here is you can return the score, so meaning what is the similarity score? It's usually between -1 and 1, 1 being the most similar. So then you can set a condition at the end where we want the score to be greater than, say, 0.9. So that gives us flexibility in terms of I'm not just returning exact matches, I'm returning those that are similar. And then in terms of determining what score you want, there are other more sophisticated ways you could do that using machine learning. So for example, you can use clustering and optimize over some sort of modularity metrics. So like how similar words are to each other versus how dissimilar they are to other words, and optimizing over that. Or for example, in this context, because we want to be overly cautious and not miss any instances, we could set a lower similarity score and just accept the fact that we're going to have to do some additional manual review to make sure that we've captured everything. All right, so interpretability is our last challenge here. So as I mentioned at the beginning, it's not enough to just know these are the finished products that we sell that contain this chemical, because ultimately we have to know how are we going to replace this. Right, so there are many additional questions that we need to answer. So one of them could be, at what stage in the genealogy of our product is the chemical used? So for example, is it used to make the drug substance versus the drug product? So that would address different people with different expertise. And where is this chemical used, right? Which sites use it? If it's only a small part of our network, then it might be a much easier solution than if it's used all across our network here. So I'm going to take a step back. Given this model, what approach would you take to try to surface that information? So as previously mentioned, we can use GraphRAG. So what does that mean? So rather than just returning the material nodes, we can return all the connected information around those material nodes. Right? So here, you can see that what we return is the path. Right? So this is the entire schema of this model here. So say we have a material node n, like 2. We have our variable relationship for ingredient 2, so we can then pass every single downstream material. And then on top of that, we can layer in information about our sites into our query. And then what we get back is not just the materials that have a similarity score greater than 0.9, but we have information on this entire schema. So for example, what material this one feeds directly into, or where this material is produced at which site. And that would ultimately answer those questions. Right? So at what stage in the genealogy of a product is this chemical used? Well, we can see what the immediate downstream material is. Right? We have a type property. So here we have that our chemical of concern is a raw material, and it feeds into a semi-finished API. So that means it's being used to make the actual drug substance. So that is very early on in the manufacturing of the product. But then here, right, we can see for this chemical of concern, what's immediately feeding into is the finished drug product. So this would be an example like red dye that may be used just as part of like the coloration of a tablet, for example. So we know here, okay, we're going to have to deal with people that work in the drug product stage as opposed to those that work in the drug substance, for example. And then where is this chemical used? This is where Graph is really helpful, because you can layer in connected information that isn't, say, just about material, but information about where these materials are produced. So here we actually see, okay, both of the chemicals of concern, they're produced at the same site. So this is actually good news, right? That means this chemical is not used all throughout the network, and maybe what we could do is look at other sites, see what they're using instead, and replace it that way. And what's great about this here is we can obviously visualize it. We can return this as a JSON and interrogate that directly. Or we could pass this JSON in as context to a large language model and then ask questions of it. So like a large language model like GPT-4o, for example, that's able to interpret JSON. We could say, ask the question, what sites produce this chemical of concern name 2? We pass in the JSON, which is reflected in the path, and we could say using only this context. And then we're grounding the LLM in our actual manufacturing data, so we reduce the chance of hallucination. So the impact of this, right? So being able to leverage graph technology, the amount of time it takes, on average, to identify chemicals of concern has reduced from days to minutes. So why is that the case? Well, a big part of the time sink is being able to just find all the relevant data, right? We have that one table, which is one source. We also have to pull in from unstructured documents at some points. By having all that information consolidated in a graph, right, we don't have to figure out where we have to look for it. And then, right, because we're able to leverage vector indexes and leverage AI, that reduces the amount of time of having to look for cases in which we overlook a particular chemical. So being able to use graph in this case has really saved us a lot of time and made our work a lot more precise. So that concludes my presentation. I'm sure maybe I'm a little early, so I'm happy to answer questions now or during the Q&A. Thank you.